Welcome. Good morning, everyone. It's great to see such a good turnout for Grand Rounds in person and on Zoom. It is my pleasure to welcome Dr. Martin Picard as a distinguished Grand Round speaker today. Martin is an associate professor of behavioral medicine at Columbia University in the Department of Psychiatry. He got his PhD in mitochondrial biology at McGill, and he did a postdoc at University of Pennsylvania with Doug Wallace, studying organelle to organism communication. His focus is linking this to the human experience with molecular and energetic processes inside the mitochondria. And it's actually mind-blowing and remarkable about his discoveries and how the mitochondria are social in nature, how they communicate within the cell and all the way to the brain and shape human experience, subjective experience, and as we'll see, mental health. He has a very vibrant lab, a large research group, the Mitochondrial Psychobiology group. They combine clinical, cellular, and computational approaches to understand how energetics and perturbations with mitochondria, including people with mitochondrial disorders, interact with key brain-body processes that both shape aging biology as well as human positive health. He is a very integrative and big thinker about tackling questions such as what is whole organism aging? How is it related to cellular aging? I have to list some of his foundational discoveries. He won't have time to mention them today. These were just years ago, but to him, they're decades ago because he moved so fast. So some of the foundational discoveries he's made include discovering that the mitochondrial behavior inside the cell follows social principles, like the precursor of social behavior between cells of the body and the social factors that operate between people. He's discovered that mitochondria build tubular structures and align their cristae. He provided the first physical evidence of non-molecular communication transfer between mitochondria, which is a foundation to thinking of mitochondria as cellular antenna and information processing network. So he has created a paradigm shift in how we view and understand how mitochondria function and work. He's also discovered that during stress, mitochondria release their DNA into the cell. And so you can measure cell-free mitochondrial DNA as an indicator of health and stress responses. This has led to the idea that mitochondrial DNA acts as a hormone, as a communication signal to help promote both adaptation and resilience. In terms of his translational discoveries, you might have read about him in the news. A few years ago, he published a paper tracking people's life events over time and examining their hair graying, and found that hair grays in dose-response fashion to stressful life events and reverses. So he was studying healthy people where the cells could actually still recreate pigment. Okay, so I will just end with that. Martin is one of the most fun and generous collaborators and mentors. And many of us here, I'm looking at my colleagues like Owen, have had the opportunity to collaborate with him over the years and continue to do so. He is a teacher of the public. He has an amazing social media education program going on and masses of followers. The world is interested in mitochondria. Your time has come. Welcome, Martin.
Well, you've heard about most of what I wanted to tell you about today. Thank you, Alyssa. It is so nice to be here. Beautiful building. First time I'm in this building. I was in the Mission Bay campus 12 years ago, I think, and this looked nothing like what it looks like today. So the growth and all of the exciting work that's happening here, I'm really inspired by. And I was really excited to be asked to come and share some of our work on mitochondrial psychobiology with you. what I want to do today is I'll show you some data and I'll show you some of the things we've measured, but mostly I want to convey some principles, bioenergetic principles. If we think about the human organism, if we think about the mind, if we think about the body from an energetic perspective, how can this help us understand and develop a more holistic and accurate picture of what health is and what we can do to support it? So this is a little summary of the kind of things that we care about, right? The ability to have cells proliferate, maintaining your stem cell pool, respond to stress responses, have neurons that fire and depolarize and communicate with each other, brain function and cognition, immune regulation, wound healing, healing physical wounds, psychological, mental wounds, and then development and aging. Every little bit of human experience and human activity requires energy. But where does that energy come from? And as you might suspect, or as you probably know, a lot of the energy that we consume come from mitochondria. And specifically in mitochondria, what happens is that there's this very elaborate chain of processes, and called the electron transport chain or the respiratory chain, where carbon substrates or disassembled and electrons are actually taken off of the carbon substrates, flown into the electron transport chain. And this quantum process of electron transfer actually leads to the production of a gradient, an electrochemical gradient. So you have this little battery that gets charged with this transfer of charges, and then the charge is used to make ATP at complex 5, as well as many other things. So mitochondria have dozens of functions. Many of us know mitochondria as an ATP producer, but it's actually an energy transformation platform that has dozens of other functions. But then you can ask, well, where does that energy come from? The energy that actually fuels a mitochondria. And as you know, it comes from the food we eat and the oxygen we breathe. So we ingest carbon substrates, we breathe in oxygen, and then those funnel into mitochondria. And the energy to make those products actually starts in plants, right? So plants have this beautiful ability to take CO2, water, and then with a few reactions, actually make the carbon stuff that we eat, the oils, the fats, the proteins, the carbohydrates, and then make the oxygen. The reaction inside the mitochondria generates, very interestingly, carbon dioxide and water. So exactly what plants need. So mitochondria close this life cycle between the plant world and the animal world. And then you might ask, well, where does that energy come from? Because energy is never created nor destroyed. So that energy comes from this nuclear reactor that sits out there in outer space. So there's quantum electrodynamic processes that happen in the sun. You have nuclear fusion, creates this energy wave, right? This immaterial form of energy beamed down as light, as photons onto chlorophyll in plants, and then this whole cascade starts. So some would say the light of consciousness comes from outer space, is materialized into chemistry, and mitochondria have the ability to unpack this chemistry back into an immaterial form of energy, which is electricity, and then this powers the human body, it powers the human mind and consciousness. So if we put this into context, right, what we're looking at here is a mitocentric view of the world. So you have mitochondria there and other organelles that are here in the middle. This is the energy transformation hub of cells, right? And we know mitochondria talk to the nucleus, where that's the passive repository of about 20,000 genes that are brought to life, right, by the energy flow from mitochondria. And the communication between the mitochondria, I won't talk about too much, but is critical for the health of the mitochondria, for the health of the cell. And it is a communication between the mitochondria that actually allows cells to then communicate with each other. And then we know that communication between neurons is the basis for cognition or memory, most likely, and for immune responses and so on. And then the ability of cells to talk to each other is actually the basis for functional organs, right? So what makes a functional brain and functional brain networks is the communication between its different components. And then we know that communication between organs is actually what makes a functional organism. So we need the brain and the heart to talk with the liver, with the kidneys, and it's the whole social ecosystem of organs within the organism that makes health and life possible. And then once you have a functional organism, then you can have people talk to each other and exchange information. So it's and then like we're doing this morning, we're exchanging information and we're creating better and greater things together in communities. So there's flow of information from mitochondria, right? This little energy transformer inside the cell, out to cells, to organs, to organisms, and then to communities. But very importantly, there's flow of information the other way around as well, right? And the forces, the social forces that we experience at the interpersonal level also ripple down to influence our brain, to influence our heart, and to influence our cells, and to influence our mitochondria. So this is what mitochondrial psychobiology is about. And one way to think about this, we need to break this down into hypothesis, into testable questions. How do we do this? So this is one model, very simple model, where you have psychosocial factors that we experience on a minute-by-minute basis, on a day-to-day basis. They shape aging biology, they shape aging physiology, and how the human organism functions. Here, many of you will recognize this as a mediation model, right? So we're asking, could it be that mitochondria and the accumulation of mitochondrial dysregulation, what we call mitochondrial allostatic load, building on the systemic allostatic load concept from Bruce McEwen, could it be that mitochondrial dysregulation or mitochondrial allostatic load actually mediates this relationship, right? That psychosocial factors and chronic stress, let's say, has effects on aging biology because it affects mitochondria. So there's a few pieces. Each arrow here is can be turned into a hypothesis that is testable. And we've been for the last few years at trying to test different facets of this model. And the idea behind mitochondrial psychobiology is that it examines the interactions between the subjective human experience, psychological states, and the molecular and energetic processes that happen in mitochondria. And there are many open questions, more open questions than answers at this point. But what I'll do today is tell you about some of the questions that we've been asking using this model. And I'll start with hair graying that Alyssa mentioned. Hair graying is a hallmark of aging. And who knows about the beautiful wheel, Hallmarks of Aging from Lopez, Orton, and others? Okay, so a few people. So this is a physiological hallmark of aging, right? Everyone who ages gets gray hair at some point, and there's a large variation of people start in their 20s. Some people start much later, but this is something that happens in humans. And one question that's been around for a long time is whether life stress is linked to hair graying. Who thinks that life stress can cause gray hair? Okay, we have a majority. It's not unanimous, but we have a majority. So there's actually not a lot of good research on this. And up until a few years ago, there was no objective quantitative evidence that life stress was actually linked to hair graying. But when you think about hair graying, there's another thing that's really striking, is on the same person. You look at a person that has a given genome, right? And that has a given experience and a given set of exposures. And on the same person, you have hairs that are dark and seemingly, youthful. And then you have hairs that are gray. So there's this heterogeneity, right? And every hair has the same genome, right? And all of these other exposures. So what makes one hair go gray when you're 30 and one hair remain dark until you're 60? If we could understand this, we thought, this could actually open some interesting ideas about what might explain the inter-individual variation in how we age and why some people age faster or slower than others. So can we learn something from the heterogeneity in hair graying? So these were the two motivating questions that initially brought us into thinking about hair graying. And so Shannon and Ayelet, two young students in a lab, started to go with this. And then what the remarkable finding at some point is that we were thinking, what if you could find a hair that's dark, right? The tip is dark, but then the root is white. So, and then you could actually pinpoint in time, if you know how quickly the hair grows, you could say, like 2 months ago, something happened and the hair that was youthful and dark actually turned old and white, right? It lost pigment. So we could situate in time life events that would correlate with hair graying. And my partner, Mary, actually found two hairs. As we were talking about this in the living room, she said, well, I've had hairs like this that are two colors. Went to the bathroom, came back with two hairs that were plucked. A segment was dark, a segment was white. I was like, whoa, okay, we have to study this. So how do you study this? So the idea is this, that you have in the hair follicle, which is this little mini organ that lives under the skin. Every hair has a little follicle. This is from which the hair grows. This is, now. And then the tip of the hair is older, right? And then as the hair grows, the same segment gets further and further away from the body. So if you don't cut your hair, you actually walk around with a bit of biological history attached to your scalp. And people have used this to study cortisol. And if you take like 3 centimeters of hair, that represents roughly 3 months of like cumulative exposure to cortisol. So and also people have used this to look at chemical exposure and toxicity and things like that. So we thought, what if you can pluck a hair, and then you can digitize it, you put it on a high resolution scanner, the same way you scan a picture in the old days, and then you could actually make a profile, right? You would track along a hair, the hair is dark, 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 and then boom, it goes white. And so this we call the hair pigmentation profile or hair pigmentation pattern. So we'd say the hair is dark and then it grays, and then you can ask what happened here. So this was the idea, and this is one of the most remarkable hair. So we started to sample people, and we asked people to send us hair in Ziploc bags through the mail. And then this was one of the most remarkable hair. What we're looking at here is the hair from an Asian 30-year-old woman. And this was beautiful because the part away from the scalp, the older region was dark, and then there was a white segment, right? That hair got old, it lost pigment, and then it regained pigmentation. So you see on the right here, the hair pigmentation pattern. The hair was dark, it grayed, and then it went back to being dark again. Full recovery. So then we asked, well, what happened in this person's life, right? So we developed a little survey to quantify this. And the survey basically asked people, take your calendar, take your phone, whatever modality you use, and then now is May, and then go back a year, right? Past 12 months, and then mark on this little chart with these grids, the most stressful time of the past year. And then for her, it was very easy. Most stressful time, least stressful time. Okay, least stressful time. And then mark notable events in the past year, 6 to 8 notable events. And then you do this. Then you connect the dots, right? Then you get this profile, retrospective profile of stress. Not great, but better than nothing. In this case, it was fairly convincing. She said, well, I was doing my PhD and it was pretty stressful. And then the most stressful 2 months of my life. Personal relationship issue, broke up with her boyfriend. Do I move to the East Coast? Do I not? She lives in California. And then there are a lot of life dilemmas, like I think many of us have experienced. Most stressful 2 months of her life. Then she moved to New York City, finds this new job, reunites with her boyfriend, stress drops. This is the stress profile aligned with the hair. So if you do a statistical test, it's highly significant, right? Something happened in her subjective experience that translated into hair biology, and in this case, hair graying, which was remarkable. So this was the first quantitative evidence of a link between life stress and hair graying. Again, retrospective, the best way to do this would be to do this prospectively. But then we were able to ask, well, what changed biologically, right? How does this happen? And then behind the back of our mind, could this have to do something to do with mitochondria? So we did proteomics, and then we contacted some proteomics scores, like proteomics on hair, forget it, especially in a single hair. But what we did was to take the single hair, chop it into pieces, and then to do proteomics on single pieces of single hairs. So this was a proteomics tour de force, I was told. It was really challenging for sure. But what we learned was that there's a bunch of proteins in the hair that were upregulated when the hair loses color, and then some proteins that were downregulated as expected. Maybe you lose color, you lose other things. But look at the proportion. There are many more proteins that were upregulated in the white hair that were downregulated. And then you can visualize this as a volcano plot. Some of you might have seen these kind of plots. On the right, if something's on the right, it means it was increased or it was upregulated in the white hair. And then on the left was decreased. What do we notice here? Many more upregulated proteins. right? So the hair loses color, yet it upregulates all these things. And then you can do functional annotation of those proteins. What are these proteins? Turns out we did this across 2 different proteomics core on a few different sets of samples. 5 proteins replicated across 2 experiments. All of those proteins were mitochondrial proteins. So in the white hair, as the stress happens to the hair, there's an upregulation of mitochondrial proteins. And this we found this at the same time as this other paper was published in Nature in mice, showing that if you activate the sympathetic nervous system, you expose mice to stress, norepinephrine actually causes hypermetabolism. It activates metabolism in the hair bulb, and this causes graying. So this started to make us think, could it be that there's something around energy metabolism and mitochondria that is actually plays a causal role in hair graying and in human aging, some kind of energy deficit. So Natalia in the lab partnered with Julien and Alyssa and started an analysis in vitro in a very simple model of cellular stress. And this was building on data from Gabriel, who was here in the audience, who was courageous enough to do a whole cellular lifespan study, a longitudinal study of cellular aging. And this is how Gabriel did this. He took cells from a few healthy people, put them in a dish, right? Each dish now becomes the surrogate for this person. And then every 5 days, passage those cells for a whole gestation period, 9 months. 9 months without missing a week, Gabriel passaged those cells for up to 42 time points. And then because this is longitudinal, every 5 days that you put your cells from one dish to the next, you can actually take a little sample, right? So it's like doing a blood draw if you were to do a longitudinal study in a person. Then you can measure how many cells are there, how quickly they're dividing or how slowly they're dividing, how big they are. You can measure the metabolic rate by putting them in this instrument. You can sample DNA, right? And then if you measure DNA methylation, telomere length, mitochondrial DNA copy number, you can measure the integrity of the genome, mitochondrial DNA deletions. You can do RNA sequencing. So you can ask, what are these cells trying to do, right? Which genetic programs are activated? Which genetic programs are being repressed? And then you can look outside of cells as secreted factors, which would be the equivalent of looking in the blood. And you can query your favorite cytokine, age-related cytokine, interleukin 6, for example, or GDF15, this interesting marker of metabolic stress, or cell-free mitochondrial DNA, which mitochondria can release and cells can release under certain contexts. But this is a cellular lifespan data set. Gabriel published this as this multi-omic longitudinal aging data set. This data set is available if anyone's interested. Gabriel created this beautiful shiny app. You go there, you pick the cell lines, the donors you want, you pick which gene you want to look at, and boom, it draws you the beautiful trajectories over time. You can download all the data, visualize correlations, do PCA, all sorts of beautiful things. So what Natalia did is she used the data from cells that aged normally under normal conditions without stress, and then cells that Gabriel exposed to chronic dexamethasone, a chronic glucocorticoid receptor agonist. So now you're asking, what's the experience of a human cell chronically being activated with the glucocorticoid receptor? What Natalia found is that these cells, even though dexamethasone or glucocorticoid stimulation is not damaging by itself, but these cells mount an adaptive or an anticipatory response to this challenge. And this is what we call a cellular allostatic load. And it involves a number of things. Cells shrink in size, they divide more slowly, they start to upregulate a number of gene programs involved in making more mitochondria called mitochondrial biogenesis. They accumulate mitochondrial DNA damage. They increase respiration. So they actually, their mitochondria are working harder and they suppress glycolysis, this other path for energy production. So there's this metabolic rewiring. And they start to secrete a bunch of things, cytokines and cell-free mitochondrial DNA, which cost energy. So what Natalia found is that glucocorticoid signaling alone increases energy expenditure by, on average, 60%. So the cells are there in the dish doing not much and normal. If anything, they're doing less because they're dividing more slowly and they're becoming senescent or quiescent, and they're burning more energy. long-term, what does this do? If a cell starts to burn energy faster, what are the consequences of this? And what Natalie found is that these cells actually age faster. And on average, 10 to 40% faster telomere erosion rate, which je analyzed. DNA methylation clocks, or the epigenetic clocks, were accelerated as well, about 30%. And a number of other features, including cell death, the cellular equivalent of mortality was accelerated, right? So the chronic stress leads to an excess energy consumption or transformation, which leads to an accelerated cellular aging. So using the cellular lifespan system, we've been able to ask a number of other questions. And one of those questions is what happens just with normal human aging? So this is a cytokine array. We're looking at secreted proteins. Some of you might know of the senescence-associated secretory phenotype, or the SASP. So these are different cytokines that are part of the SASP. These are things that older cells start to make more of, and they starts to spit out. What we're looking at here in this heat map, these are the young cells from donor one. So there's one person, and then the first time point, and then the second time point, third time point, and so on, for up to 258 days. Kudos, Gabriel, for your courage to do that. And then the color marks the level of the cytokine, how much there is. So notice here, this is the scale. If you go from -5 to +5, this is a 10 unit on a log2 scale. That's 1,000-fold. So there are proteins virtually that young cells don't make. Young cells have a very nicely contained epigenome, and the chromatin, and some genes are very solidly repressed, and they're not expressed uselessly. And then as cells age, there's epigenetic drift and transcriptional drift, and then some proteins randomly start to be made and can be upregulated up to 1,000-fold. So what we see here is this progression from low levels of protein to very high levels of protein across lifespan. Then Gabriel repeated this in 2 other donors, where you see the same kind of pattern of age-related upregulation. So some proteins upregulate by more than 30-fold across the lifespan. This is here the validation of the specific protein, GDF15. I'm curious to know who here has heard about GDF15 before? Few people. It is, at the moment, I think the most studied protein in the biomedical sciences. There's, if you set yourself a PubMed alert for GDF15, every day you get an email. So it's the best marker of human aging in terms of single protein. It's very highly upregulated in the blood. It's a predictor of mortality. It's a predictor of cardiovascular events. It's associated with a number of different diseases. In cultured human cells in a dish where you isolate one thing, the passage of time, you see this exponential increase in GDF15 that peaks around 90-fold on average across people. Cells also start to secrete cell-free mitochondrial DNA, which we suspect now are actually cell-free mitochondria. So cells are actually spitting out their mitochondria and spitting out mitochondrial DNA, potentially for signaling purposes. This increases 10-fold. We're not talking 50% or 1-fold, 2-fold increase, 10-fold. The cost of building mitochondria, the energetic cost of building mitochondria, replicating DNA and then spitting it out is high, and as well as making proteins. So what this ends up doing, Gabriel quantified here the amount of energy that cells are burning per unit of cell. This is the equivalent of basal metabolic rate. How much energy is the human body burning per kilogram or per pound of body weight? This is the equivalent at the cellular level. And you see also this exponential increase that is on average 5-fold from young days, early days to end of life. But during this process, cells are actually not dividing, right? And some of you may know this from a Hayflick, the Hayflick limit experiments from many years ago, where Leonard Hayflick took human cells and passaged them in the dish, and then at some point they stop dividing, and then they go senescent or quiescent. Right? So cells are not dividing, yet they're burning way more energy. What are they doing? Seems like they're spending all of this energy producing proteins. So senescence and quiescence come at a cost. There's a lot of energy that goes into this building this and maintaining this senescence program. So I think most of us has senescence and quiescence in mind, meaning like you go dormant, like a bear hibernating or something like this. So those terms are misnomers. If you look under the hood, if you you peel the plasma membrane of the cell, you look inside the cell, there's a lot more going on, even though on the outside, the cells just look like they're staying there and doing nothing. Just like if you're in front of your computer, you get this threatening email that your grant was rejected or something like this. And you say they're composed, inside your heart is beating and your hormones are going, this hypermetabolic state. So why do aging cells become hypermetabolic and start to burn energy so much faster? There's a number of things that happen, and I won't go into the details here, but cells accumulate damage. And most aging scientists agree to say that damage accumulation is a major driving force for cellular aging. Right? So you have mutations that accumulate in the DNA. There are chunks of DNA that start to jump called the retrotransposons. Chunks of the mitochondrial DNA can be translocated and then integrated in like a piece of viral DNA into the nuclear genome. So there are all of these things that destabilize aging cells. Over time, these are stochastic, entropic processes. This triggers stress responses. So cells are not blind or insensitive to this. They actually mount stress responses as they accumulate damage. Those stress responses trigger energetically demanding stress responses, like protein secretion that I was just mentioning. The SASP is a very energetically expensive process. Release of cell-free mitochondrial DNA, transcriptional noise, and expressing genes that used to be repressed will cost energy. Making more mitochondria will also increase the energetic cost of the cell, and then reshaping metabolism from using your mitochondria to using glycolysis. There are inefficiencies there. All of this converges towards hypermetabolism. So it is the stress responses in the aging cell that we think is a driver of the cumulative energetic cost of cellular aging. And senescence and quiescence might actually be compensation, as opposed to being the driver of these phenotypes. Stopping cell division might just be because cells are running out of energy and they can't afford to divide. So aging cells become hypermetabolic, that's quite clear, but the whole body does not. Right? So if you think about an aging organism, there are cells here and there that are scattered pretty sparsely that are becoming hypermetabolic. Yet if you look at an older individual, the whole body does not become hypermetabolic. This is the best data we have of human energy expenditure across a human lifespan, from 0 to 100. You see this beautiful blip here as the developing childs are growing and developing and building personalities and wiring their brain and learning about the world. There's this beautiful, the highest level of energy expenditure is in childhood, which might be the basis for vulnerability and during these critical periods of development. But and something else to notice here, those people that are above here, right, they're burning energy faster. This is basal energy expenditure. How much energy you're burning based on relative to what's predicted for your gender, sex, and your body weight. If you burn energy faster than you should, there's a lot of good data showing, as in cells that become hypermetabolic die earlier. People that are hypermetabolic also die earlier for reasons that aren't quite understood. But the whole body does not become hypermetabolic. It actually declines at the end of life, right? So you have accumulating cells that are hypermetabolic here and there in your organs, yet the whole body becomes hypermetabolic. This creates kind of a question. And this is just more data showing that with advancing ages from BLSA, the Baltimore Longitudinal Study of Aging by Luigi Ferrucci, and if anything, basal metabolic rate declines in females and in males. And this is just more recent data showing the same thing in over 1,000 people. So cells become hypermetabolic, the whole body doesn't. What's happening? This seems like an energetic paradox, right? How can this happen? And we think that this paradox can be resolved by putting the brain in the picture, right? If we stop to think about what we're actually looking at is not a bunch of cells that are just glued together, right? This is a cell collective that obeys, I think, what looks like social principles and that are regulated by an integrating principle that the brain is kind of in charge to mediate. So one way to resolve this is to think about the effect of these cellular hallmarks of aging and the cellular hypermetabolism, the molecular imperfections, the molecular damage that I mentioned, cell senescence. This triggers signals right? The senescence-associated secretory phenotype, inflammaging, maybe some of you have heard about this, metabolites, other factors. The brain has evolved to know about what's happening in the body. And just like when you're mounting an immune response, the immune system makes cytokines, the brain feels those cytokines, and then triggers what's called sickness behavior, right? You feel sluggish, you want to be in bed, conserve energy for your immune system. So here's similar idea. Aging senescent cells are producing those signals, reaching the brain, and then the brain says, whoa, I'm going to run into an energy deficit if I keep going like this. There's something in the body that's burning so much energy. Let me conserve energy. So this is the conservation, energy conservation responses. And the brain is able, we know this very well, to produce affective states, right? To make you feel uninterested in life and in others, to drive sedentary behavior and inactivity, to suppress the production of costly hormones, growth hormones, thyroid hormones, testosterone, and to turn down physiology so that it limits the energetic expenses, and also to blunt your sensory capacities and to produce other deficits. So the manifestations of aging, as we know it, and what at the extreme we call frailty, could be brain-driven processes. And I'll just say a bit more about this. So this is a bit more of a detailed view of how we think this is happening. So you have aging cells that are accumulating damage. They're activating stress responses, like I mentioned earlier, like the integrated stress response, the ISR, producing cytokines and metabokines, which can trigger immune activation, which can further increase this. Cells, as I mentioned, will release mitochondrial DNA, which might activate some DNA sensing pathways, and there's some complexity there that might create a positive feedback loop. This leads to the release of cytokines and metabokines and chemokines in the circulation in the blood, which we know increase with human aging. Those are sent by the brain. The brain has this beautiful, these elaborate structures that are covered by receptors for cytokines, so that the brain can know what's happening in the periphery. And then there's this elaborate neural circuitry that's involved in integrating those signals. In turn, then the brain produces outputs, neural outputs, endocrine outputs that can affect systemic metabolism and shape behavior. And then in turn, those chronically changes in metabolism, changes in behaviors will lead to anatomical changes, right? The brain can shrink your muscles by decreasing physical activity and reducing activation of the neuromuscular units. The brain won't shrink muscles, which is a very good thing to do if you're running into an energetic deficit. Shrink your brain, right? People can live with much smaller brains than they actually need to. So that's another way of saving costs. And there's a number of things the brain can also do, simplifying the immune system, suppressing immunity, for example. So all sorts of things that we associate with bad aging consequences might be driven by the brain. And the product of this is decreased whole body energy expenditure. So we have the situation here where we have cellular hypermetabolism leading to whole body hypometabolism. I'll just skip this, which was a summary of the brain-body energy conservation signature. The key point here is that it's an organelle to organism bioenergetic principles that coordinate this communication from aging senescent cells to the brain. And with an understanding like this, I think we're better positioned to make good decisions about what kind of interventions are likely to be useful in the short term as well as in the long term. So the brain is an important piece in this puzzle here. Turns out the brain is packed with mitochondria. And it's one of the most energetically expensive organ in the body. And one question that has not been resolved to this date is, how are mitochondria distributed and how do they specialize across the brain? The brain has an enormous energy budget, and it's constant. So on the left here, you have the size, right? The relative mass of different organs in the body. You see here the skeletal muscles, very big chunk of the human body. Fat for most people is a fair chunk as well, other tissues. This is the brain, the little blue sliver here, about 2% of body weight. So relatively speaking, the brain is pretty tiny in the whole body. This is the fraction of the energy that the brain burns per minute, per hour. The brain burns 20 to 25% of the whole body energy budget, despite its very small size. And interestingly, there are things like skeletal muscle. If you're sleeping or if you're sitting passively listening to a talk, your muscles aren't burning too much energy. But if you start to run, or you're trying to run for the bus, avoiding the rain in the morning, then the muscle energy expenditure can actually increase 10 to 20 fold. The brain, and not brain changes in energy consumption, actually is in a few percent. It's rarely above 5%. So there's something in the brain that's costing a lot of energy. And I think the debate is still out as to what is it about this and about sustaining consciousness or other processes that's costing so much energy. So that energy is coming from mitochondria. And we know this very well. If you stop breathing, or if you occlude blood flow to your brain, 5 seconds later, you're out, right? The flow of energy through mitochondria is directly linked to your consciousness. So there's something critical there. So how do we figure out how mitochondria are distributed in the human brain? This is a really challenging problem for a few reasons. One, the brain at the scale of mitochondrial biology operates at the scale of cells, and at the micro scale, the brain is a macroscopic organ, and it's beautiful. This is a picture here of a human brain. This is a single section of the human brain. And what we wanted to do is to create a map of mitochondria across the brain. So we call this a mitobrain map version 1.0. The goal was to create a multifunctional mitochondrial atlas of a single human coronal section at fMRI resolution, right? So we can actually bring this to bear. This was a huge team effort. This was 3 years of work led by Ayelet, and student I mentioned earlier, and Eugene, who's a scientist whose office is right next to mine, he thinks about life like an engineer, but he's a chemist by training, and he works in a neuroscience lab. So you bring these things together, and then with a bit of mitochondrial infusion, and then this can happen. So the goal was to close the gap between organellar bioenergetic profiling and the whole brain neuroimaging modalities, right? These are two completely different worlds. People who do cognitive neuroscience never think about what happens inside the cell, and people who look at things inside the cell never think about what happens in a whole organ. So what Eugene did was to take this beautiful, look at how beautiful this is. This is, some of you might know, but the white stuff is the white matter. These are axonal tracks that can be, multiple centimeters, inches long. This is here the putamen. Here's the corpus callosum that connects it to hemispheres. The brown stuff here is a gray matter. That is a cortex. This is the hippocampus, the beloved hippocampus that is involved in memory and Alzheimer's and so on. So all of these beautiful areas in the same organ. How the hell do you partition this in a way that you can do mitochondrial science on this? So Eugene thought, well, let's order a CNC cutter, a computerized cutter, like doing, if you want to engrave a heart on a piece of wood for your mom for her birthday, right? You use these kind of machines. So Eugene programmed a CNC cutter to engrave a grid. Right? He's doing horizontal lines now. He's doing vertical lines. And then he thought, if we do this and we engrave 3 millimeters deep, then we could actually turn the brain into 3 by 3 by 3 millimeter cubes. And if we do this, this is the standard resolution for neuroimaging. So then you can, and in neuroimaging, people speak in terms of voxels, right? So the MRI actually turns the whole brain into little voxels that are 3 by 3 by 3 or 2 by 2 by 2. So this is physical human brain voxels. And this is what they look like from up close. It's that little cube there. And Ayelet and Snehal went at this for hours. And then one voxel by one, pluck it out, record it, register it. And then once you have these little cubes, these little samples, you can actually run biology on them. So as expected, each voxel 3 by 3 by 3 should be 27 milligrams. So you get this beautiful distribution peaking at 27 milligrams. The edges of the brain are a little smaller. If you look at protein abundance, gray matter, white matter seem to be roughly have the same protein abundance. This was kind of quality control. So we've turned the brain in the slice section of brain into 703 pieces of brain. Now, can we do mitochondrial biology in this? So we've miniaturized some mitochondrial assays in the lab that we've called mitochondrial health index that we've used before in a study with Alyssa. So now we could do the mitochondrial health index, the enzyme assays on these brain samples. And we partnered with colleagues at UCLA to do a different kind of mitochondrial profiling method on the same samples, bring those two together for additional robustness, and then you get mitochondrial profiling in all of these voxels. And now put this into the context of the brain. So what you're looking at here is 3 years of work to profile the mitochondrial activities. The heat map yellow and orange is higher activity, black is lower activity. So roughly you see that the cortex, right, the outside of the brain, where there's a lot of cell bodies, lots of synapses, tend to have more mitochondria, but not only more mitochondria that are specialized for energy transformation. And what we're looking at here is OXPHOS specialization, oxidative phosphorylation. You can have a mitochondrion that's specialized for making hormones, like in adrenal glands, for example, or you have mitochondria that are specialized for transforming energy with the electron transport chain. This is a map of the specialization of mitochondria across the human brain. And you can quantify different brain areas. I won't go into the details here, but compared to the white matter, right, those white matter tracks, these axons, they have very few mitochondria that are not specialized for energy transformation, compared to this most cortical areas, and some cortical areas more than others are particularly specialized for energy transformation. You can project all of this data into lower dimensionality reduction space like UMAP, which people use for single cell analysis. And what you see is that there's distribution, non-random distribution of mitochondrial properties. And then we wanted to bring this at the scale of the whole brain, right? So we profiled, it took 3 years to profile a single section of the human brain. How do we bring that at the scale of the whole brain? And we did the math. It would cost a lot. It would take a lot of time to do the whole human brain. So one approach that we decided to take instead is to use neuroimaging data. And we registered our human brain section into standard MNI space, the Montreal Neurological Institute space that people in the neuroimaging world use. So now you have this brain morphed into a standard neuroanatomical space. There are thousands, tens of thousands of people and data set for neuroimaging that we were able to bring together. So now we can ask, using standard neuroimaging modalities like fMRI bold, which looks at blood oxygenation, T1, T2, just structural brain scans, or diffusion imaging to do tractography and to look at white matter. Can we use that information, average information across hundreds of people, map this onto the mitochondrial brain map, and then build a predictive model? So we used 80% of the 703 voxels, train the model, and then try to predict on new voxels, unseen voxels from the model. This is the prediction accuracy. You see observed here, that's what we measured. This is predicted, R-square of 0.36. I'm told by my neuroimaging colleagues, this is pretty good. If you're comparing one brain from an average from hundreds of brains, it's hard to expect better than this. And then if you look at different brain areas, this is collapsing multiple voxels that belong to the hippocampus, for example, or to the frontal gyrus or medial temporal gyrus. You see that the prediction is quite strong. And then we had from the same brain of the occipital, a different area that we didn't initially use in the model. And there's also kind of internal validation of the model there. So what this allowed us to do is to create a whole brain probabilistic map, right? Using real measured mitochondrial biology in one section, you can predict the whole human brain. And this is the predicted map of mitochondrial respiratory capacity, the second generation of the mitochondrial health index across the cortical surface here. So many questions now that we have this resource, many questions we can ask about large-scale brain networks, a default mode network, the salience network, and all sorts of cool things that neuroscience, neuroimaging people are interested in. So I'll finish by talking about one analysis that Caroline Trumpf, a postdoc, who was a postdoc in our lab and now is a faculty and leading an exciting research program, building mitochondrial psychobiology to the world epidemiology. And what Caroline wanted to ask is whether brain mitochondrial phenotypes are linked with psychosocial exposures and experiences in people. How do you do this in people? So what Caroline did is she leveraged the ROS map cohort, which is based out of Chicago. And it enrolls participants age 65 and over, and then every year follows them up. So every year you get a measure of psychological well-being, of depressive symptoms, of anxiety, of social network size, and other contextual psychosocial factors, positive and negative. And then at the end, when people die, half of the hemisphere goes for analyzing Alzheimer's disease pathology, and the other half is frozen for molecular analysis. On the molecular analysis, what was done by the ROSMAP team is to measure a number of things, including proteomics and bulk RNA-seq, and now they've done single nucleus RNA-seq. So there's deep molecular multi-omic profiling of these brains. And then this is the design. This is death here. Post-mortem, you get the brain. You can do all of these interesting measures. And then antemortem or pre-mortem, then you get positive and negative psychosocial experiences. So then that allowed Caroline to ask, okay, can I correlate? Is there any association between what people reported experiencing before they died and then their brain mitochondrial biology after they died? This is what Caroline found. Looking at the association between psychosocial, positive, negative psychosocial experiences, you could think people who report more positive experiences probably report more less negative experiences. There's some collinearity between those, but not completely. And then if you look at the correlation between the composite score and mitochondrial complex 1 abundance, complex 1 is the first protein that brings electron into the electron transport chain to build the life sustaining gradient in mitochondria. There's a significant correlation. The proportion of shared variance here is 18%. R square, 18%. So 18% of the variance in the abundance of complex mitochondrial complex 1 across people's brain based on this analysis could be explained by psychosocial exposures. Or if you have more complex 1 protein in your brain, maybe you perceive the world in a more positive way, right? So we can't know what direction this is coming from, but this association is intriguing. There's also single nucleus RNA-seq or single cell analysis in those brains, and Caroline did an analysis that I won't describe now, but she looked at this question, right? Is there a cell type specific association between psychosocial exposures and certain brain cells? So this signal where we're seeing 18% of shared variance, is this because the neurons respond more to psychosocial exposures or more strongly related to psychosocial experiences? Or is it the oligodendrocytes? Or is it the astrocytes? Or the beloved microglia, right? Which cell is it? And it turns out the glial cells is where the action is at. So this is a project that's still in development, but there are cell type specific associations between psychosocial factors and mitochondrial oxphos. Not every cell seems to be related in the same way to what people are experiencing. So this is overall, as I said earlier, the model that we've used to guide our inquiry into these questions, right? We've asked questions about the effect of psychosocial factors on mitochondria, in immune cells. I didn't talk about this. In the human brain, I just showed you an example of a biological question in mitochondria. We know if you perturb the mitochondria, you perturb cellular aging. We know this from Gabriel's cellular lifespan study. If we apply stress factors, we accelerate cellular aging. So we're starting to test a few of those arrows and to understand some of the energetic principles that might be guiding cellular resilience or cellular vulnerability to stressors. I want to come back to this picture. This is, I think, the most accurate, the least precise, but the most accurate picture of how the whole system is wired. And intrinsically, it's wired to respond to energetic signals, right? Cells naturally will respond and choose to divide or not divide or die based on the energetic signals they receive. Function of the brain is regulated by energetic factors and up to social behaviors. And there's beautiful evidence in animal models showing that if you perturb the mitochondria, you actually change things like sociability or social dominance and these kind of complex behaviors. We know now we're subject to energy. So energy doesn't just play a life-sustaining role, is actually plays an instructive role in those processes. And very importantly, again, there's flow of information downwards. And the stuff that we experience on a daily basis, the environments we create for ourselves, the relationships we are embedded in and that we experience end up trickling down probably all the way to mitochondria, and more work is needed to understand how this happens. So thank you so much for I just want to say this is our team who's doing the work now that I presented and some of that I didn't. Here's Caroline, who's co-leading our team and supervising some of our lab members. And here's Eugene, who did the mito brain map, brain voxelization. We have several precious collaborators that make this work possible, and I'm so grateful for. In particular, I want to thank the Bazooki group that's been supporting us in expanding towards exciting projects that our NIH grants can't support. So thank you so much again for your attention.
Thank you so much. We have 5 minutes for questions. Any questions from the audience? The Bazooki group just awarded Martin a $1.5 million prize for his amazing research. And I guess I also forgot to mention, he has 8 R01s. I don't know anyone. So he's a very busy person. And so anyway, we have a question from the Zoom room. So first we'll have Rob, and then we'll have our question.
All right. Martin, a real tour de force.
Thank you.
2 things that have come out recently regarding mitochondria and energetics and cell aging, cell death, are the fact that mitochondria seem to split, and when they split, they don't have nearly the oxidative phosphorylation capacity.
Mm-hmm.
And the other is Bob Navio's work about extracellular ATP actually leading to cell damage and being a marker for cell damage. It would seem to me that mitochondrial DNA promoting hypermetabolism versus extracellular ATP looking at cell damage might be an appropriate marker for what's actually going on. Have you looked at either of these 2 phenomena and whether or not this then explains what's going on and gives you, cause to go backwards as to what the source of that is?
Yes, thank you. Good question. So the question was twofold: one about mitochondrial dynamics and mitochondrial fission and fragmentation, and then about ATP, not just as an energy currency, but also as a signaling factor. We've done some work looking at the mitochondrial morphology and how this relates to cellular, this energetic state and stressors. I didn't show any data here. Others have done a lot of work showing that the shape changes actually precede and are required for functional changes in mitochondria. So the function of mitochondria is related to their morphology, and their morphology is one aspect of their behavior, of their social behavior, that is regulated in the minute time frame. So you can look at a cell under a microscope and you see the mitochondria move and change shape and respond to all sorts of beautiful signals. And the fact that ATP is a signal, right? That intercellular cells talk to each other by secreting ATP and sensing ATP, I think says a lot about the need for the organism to be tuned to energetic signals. So ATP is both an energetic signal and probably plays a lot of other roles between cells and systemically in the blood. We haven't looked at this specifically, but certainly something that we should look at.
One last quick question from the Zoom audience is, can mitochondrial biology help explain the positive effects of fasting?
Yes. So there are 2 things we know really well that helps promote lifespan and health span is 1, moving, being physically active, and 2, not eating too much. And there's a lot of interest for understanding how fasting, intermittent fasting, and maybe some version of calorie restriction has positive health benefits. What seems to happen in cultured cells, if you do fasting on cells, is the mitochondria right away become more social, or it promotes fusion. So mitochondria that are challenged or in difficulty can actually fuse with a good, healthy, robust mitochondria, and then now they can support each other. So it seems to, I think our interpretation is that it puts the organism in a state that forces collaboration and partnerships. And you see this at the level of mitochondria fusing with each other. And there's good evidence that there's also signals between cells that are being triggered by fasting.
Yeah. Amazing answer. Thank you so much, Martin. Martin has a few minutes. If you have a question, you can come and visit him. Thank you.
Thank you.